Okay. Hi, everyone. Hi, how are we? Scale of 1 to 10, how do we feel about this final? 8.
Oh,
Pretty good. Okay, 7. Wow, very confident. Okay, anyways, so if you're watching this video later, or if you're here, this is the final exam review session for Bio 285. Sorry, one sec. I just need to make sure that the sound is. Sorry. Okay. Okay, sorry. Just making sure that the audio is clear. So this is the final exam review session for Biology 285. This is one of 3 review sessions, so we're just gonna get started. A few announcements. First thing, congrats on reaching the end of classes. You guys did a good job powering through this semester. I know besides this class, you guys have plenty of other things to take, probably Orgo and all that, right? So give yourselves a pat on the back for making it through that. There are 3 final exam review sessions total. You didn't see like all my 8 emails I sent. There's 3 happening. So this is one of the 3. And just make sure to tune in to your emails for the times and locations for that. When is your final? If you didn't know, your final is December 15th, which is a Wednesday, right? Wednesday, okay, great. And your final on Spire, I think it's scheduled for 10:30 to 12:30, right? Is that the time? Sometimes it changes, so I wanna make sure. But if you guys know, when from experiences with taking her exam, sometimes you might need a little more time. And if that were the case for you, do not panic. You do have an option to be relocated to a different area to be able to continue your final, because your final exam will be in Topman Gym, but most likely there could be another class that needs to use the area to take the final. So what Laura did in my year, and Zayed can speak to that, is that we all walked from Topman with our exams to like a different area to finish the exams to continue. So most likely Laura will have a backup space set up for 12:30 and onward for people who still need to work on the exam. Any questions on that? Or does that make sense? Yep, question?
Do you know what percentage of the exam is multiple choice?
Ooh, I'm not sure. I haven't heard word about what percentage is which, but I think it depends on how many TAs can grade the exam. But I don't want to say definitively for sure or not for sure, but please email and we can double check. But I think there may be some multiple choice included on yours. As terms of the percentage, I don't know how much. Sorry.
What if
But-
half the TAs get covered?
Okay, good question. We'll save it for later. Okay, next thing. Your exam, you get 3. Sorry, I'll be there. Your exam, Laura said that you can bring the cheat sheets from prior exams to your exam. So you're welcome to do that. And then an additional cheat sheet for weeks 11 through 14. So yes, please do that. And she said on your exam, I think she said in the email that you will get nuclear import as a diagram attached to your exam. And there's another diagram, but I'm forgetting. But other than that, just make sure your cheat sheet is filled with information and diagrams that you'll need to be able to answer the questions, which it already should have if you used it for like pathways and stuff like that for exam 2. And you'll just know things that you'll need to put on there. Yeah, you had a question back there?
Yeah, I have an exam like right after this one.
Mm-hmm.
Like what if I don't finish in time for like that period? I feel like I broke one just in case.
Okay, so I wouldn't operate on the probability of chance. So please make sure you reach out to Laura and let her know your situation. She's very accommodating.
Cool.
For some, if you have a conflicting exam or things of that nature, sometimes the other professor might be like, you have to take my exam this day, this time. So there's a way you could probably work around that. Whereas if the professor's not letting you switch the exam that's after Laura's, then Laura might be like, oh, maybe you can take it at an alternative time. So she works with people to make sure that they're all able to take the final, because that's important. It's worth 100 points of 514 points. So it's important. But yeah, she'll make sure you take the final. But it's really, I wouldn't leave it up to chance. Just communicate, please, with the professor, because we don't want people panicking to finish the exam to go to a next one. If you could have had, if you need more time to work through her exam, because it takes time to think things through. So that she could have alternative time slots. And I know you're not the only one who has that. I'm sure plenty of people have exams the same day. and she'll have most likely alternative time slots. But you need to email her to communicate that, otherwise she wouldn't know. Okay, any questions? No more questions? Okay, and then what else was I gonna say? These review sessions are great, but I don't really want people to just use only the review session as a way to study for the final. You guys know that. Just please make sure that you look through your notes and everything for all the material, because our review sessions, we can't include every single thing that you guys have studied in the semester. We just highlight a lot of the important stuff. So we don't have all the nitty gritty details on exactly everything, but your notes and all your class resources, you will find those things. So definitely like utilize the review sessions as a resource, but don't just only rely on these to help you with the final, please. Please study. Thank you. And yeah, you guys know that the TAs have been giving you excellent study habits for every exam review session. If you didn't know, now you know. You can look on YouTube for those. And in the emails and the slides, we include a lot of active learning study habits. So please read through those. And something that I thought might be useful to share is that Dr. Francis makes new questions that are similar, but not identical to old exam or problem set questions. So maybe you should spend some time studying those and understanding why an answer is a particular answer, explaining your reasoning for those. So yeah, big thing to understand why you got an answer wrong and the explanation behind it, behind the correct answer is key. You can even do this when you're studying multiple choice questions. You can say, okay, the answer is C, but why is the answer C? Or why isn't the answer A, B, D, that kind of thing. And yes, if people didn't know, on Moodle, there's this thing right now, forward focus survey. It's, I don't know if you call that, I think it's extra credit for them to do it. 5 whole points added to your entire total for this class. If you don't do this survey, okay? Because you don't want to be like deprived of points. And you don't want to be like emailing, please open the survey, I need to do it. It's like extra credit towards your grade. It's gonna help you overall in the class. That those points could be a difference between like different grades, you know, so definitely do it. That's why I put it in the slide, because we want everybody to get their points. Does that make sense? I'm not sure the deadline for that, but if you haven't done it, please do it. Any questions? No? Okay. Okay, and these were just the tips I included for active learning, but I always say these tips every time, so let's just skip that. Okay, this part was important. I said this in exam 2 review, and I'll say it again because it matters. Wait, why are these? Anyway, so you have this thing, special magical thing called final exam substitution policy. What is this thing? If you didn't listen to my YouTube videos earlier or see my slides, I'll explain again. Basically, final exam substitution policy means is that Laura will replace your exam 1 or 2 midterm if your final exam beats the lowest grade. I feel like it didn't make sense when I said it, but let's look at an example. So basically, if you do really well in your final and you beat your lowest midterm score, your final kind of counts like twice in a way and like negates that lower midterm score. Does that make sense? So we got an example here. Student A, or X or Y, Z, whatever. So student, exam 1, they got an 80. Exam 2, they got a 78. But on the final, they got a 75. So what does that mean? No change in their grade. Their final exam stays at 75 out of 100, and then the other one would be 39 out of 50, or right, 39 out of 50, which I think 78%. So that's just an example of that not working. Now, student B gets redemption. Why? Because on their first exam, they got a 73. Exam 2, they got an 80. But on the final, they got an 85. And so that would mean for student B, their final would be an 85%, and then for their exam 1, it gets substituted to an 85% as well, to reward the student for understanding the material on the final exam and taking away ideas from the class. That's why she does this final exam substitution policy, is to reward you for, you know, your efforts and working hard to study for the final. And if you understood the final and you did better than in the midterm, it just shows that you like learned from that. And that's why she does the substitution policy. And she usually does that with a spreadsheet so she can tell like the scores and everything. But if there's any discrepancies in Moodle, you can email her and let her know. Yeah, so the idea behind that, she always does it every semester. And one bad exam grade in this class is it shouldn't be game over. I don't know if you guys ever taken certain classes that you do a bad score on the exam and like you feel like, oh no, like what's the point in even like doing this class or trying? And I think she just really tries to emphasize that you should like keep trying, and that like one bad grade is not going to cost you, however you define like bad for you. in that regard. So yeah, and that's just her final exam substitution policy. And this student would be an example of someone who would get what I call redemption with that policy. And just keep in mind this class, a lot of classes usually are like percentage based for components, but this class is a points based system. You guys can consult the syllabus for that, but your exams are worth like 200 possible points. And so there's a way to calculate your point score and all of that, but that's just the way the class is. Anybody have any questions about that? No, no questions? Okay, go
Is
ahead.
the max 519 with the quiz or sorry, the survey, or does it stay 514?
The maximum grade you could get in this class, I believe, would be 514 out of 514, I think. But the survey just like adds points to your like already total. So after you took the final and everything, whatever your points total is, divided by 514. Like there's a whole like syllabus thing to it that's in the syllabus. But I think the survey Ford Focus, it doesn't get added to an exam score, it's to the grade overall. Okay, does that help with what you're asking?
Yeah, I was just asking if it changes the total amount. That's all.
No, your total would not be like bigger because you did the survey. No, I don't think so. But you could email her to double check, but I don't think so.
Should be like say you have like a 500 out of 514, then like if you did the Ford Focus survey, you
505. Yeah,
Yeah.
what Tina said, it just adds to your overall total. So you're not like gonna, in a way, you don't lose points by like not doing the survey. It's just like, that's why I call it extra credit. So does that make sense? Is that a question? Just stretch. Okay, sorry, I was just like looking out for questions. Okay, so now to get into the 285 stuff in here. Content being covered today, weeks 1 through 4. Okay, cool. So we can get started.
Okay, so we're gonna start with week 1 stuff, which is amino acid and levels of protein structure. So pretty much we have, starting off with our primary structure, right? There's the primary, secondary, tertiary, quaternary. Your primary is just gonna be simply the sequence of your amino acids. And it's gonna be formed by the peptide bonds that are gonna link them together. And the important thing to know for this is that these are gonna be covalent bonds, right? So they're gonna be your stronger, more stable bonds. And that makes sense because if you lose the primary structure of a protein, then the entire thing just falls apart, right? Because this primary structure is what's going to influence the secondary structure, which is composed of your alpha helices, beta sheets, and it's also going to determine the interactions in your tertiary and quaternary structure. So again, it's just a sequence of amino acids. It goes from your amino end, which is your nitrogen and hydrogen on one side, and then it goes to the carboxylic end on the other side, which is your carbon double bond oxygen. And yeah, so pretty much these are just formed by the peptide bonds. You can see it goes in the pattern of NCCN. So if that's what you see, that's how you know you're looking at the primary structure, because that's like the abbreviation, right? You don't see any R groups or anything like that. And just know that these peptide bonds are very strong and they require a lot of energy to break. And then you have your secondary structure, like we said. This is the formation of your alpha helices or beta sheets. And this is pretty much just caused by the hydrogen bonding you can see between the hydrogen of the amino group and the oxygen of the carboxyl group. So pretty much the orientation of how these bonds are formed is going to determine whether or not it's an alpha helix, which is on the top, or a beta plated sheet. And pretty much the only difference that you need to know is that for your alpha helices, you're going to have your R groups orientated towards the outside. So you're going to be having your interactions be formed with whatever solvent your protein is in, versus your beta plate sheets where it's going to be interspersed. So you're going to have some R groups on the outside and some on the inside. And think how like that would make sense for versus if you have a protein with hydrophobic parts and hydrophilic parts, you would want to have a beta plate sheet, right? So you can have both interactions. Or versus if you have an amino acid sequence of just hydrophilic and it's in water, say for example, it's in water, you're going to have your alpha helix. And other things, yeah, so these bonds are going to be called your backbone atoms because again, they're only going to be made by the amino groups or the carboxyl groups. They're going to be dipoles because there's a partial charge, right? You have you're interacting with hydrogen off the oxygen, which is very electronegative, and the nitrogen, which is also partially electronegative. And the last thing is pretty much these are going to be non-covalent interactions. So not as strong as a primary, which pretty much tells you that it's easier to denature this level of protein structure. Any questions? Okay, so, and then we have your tertiary structure. And this is going to be the actual like 3D shape, like the physical folded product of your protein. And the important thing to know is your tertiary structure is only going to be a single polypeptide chain. So just one chain of amino acids is going to be, after that folds, depending on whatever R groups it has, it's going to be your tertiary structure. And the interactions are going to be all non-covalent, but they're going to be hydrophobic interactions, your van der Waals, ionic bonds, hydrogen, disulfide bridges, which we'll get into later in the review. And then you can see there like how it folds, depending on if there's a disulfide bridge or if there's hydrophobic interactions. And that's where the importance of R groups comes in. So we have our quaternary structure, and this is going to be any two subunits or more and their interactions between their backbone and their R group. So interactions between any two long polypeptide chains in their secondary or tertiary structure. And for example, we have a hemoglobin protein, which is made up of 4 subunits, 2 beta subunits on top and 2 alpha subunits. And you can see by the color coding, you can see how they're made up of 4 individual polypeptide chains. So then some differences between tertiary and quaternary. Again, tertiary is just one polypeptide chain and the way it folds. Quaternary would be any 2 or more polypeptide chains together. And those then would be called subunits. So if you have a quaternary structure, whatever one polypeptide is going to be one subunit. And on that subunit or on that one chain is where you're going to have domains of different R groups and how they interact. So does that make sense? The difference between quaternary subunits and then tertiary and domains. Is that okay? And then oh yeah, and then and then again, these are going to be stabilized by your covalent non-covalent interactions. So your hydrogen bonds, electrostatic, van der Waals, and then any more such as like disulfide bridges and such. So, oh, and again, like not all proteins are made up of multiple subunits. You can have a full protein with just one tertiary structure or one polypeptide. And again, this is just a diagram of the different levels, right? You start off with your primary structure, the individual amino acids, and then you have your secondary structure. In this case, we have an alpha helix. And remember that in this case, the R groups will be facing towards the outside. And then you have the tertiary structure, which is that one helix folded on itself, depending on whatever R groups it has. And then you have a quaternary structure, which is, say, any other polypeptide chains that come in and then make interactions between those secondary and tertiary levels. So is that good?
I was just going to add, sometimes I think it's helpful to look at it from starting with quaternary structure and zooming out. So if you look at quaternary structure, you have the 4 subunits, then no, I'm sorry, what am I saying? Zooming in, sorry. If you have a quaternary structure with the 4 subunits, then you zoom in on one of the subunits, you have tertiary structure. Then you zoom in more on tertiary structure, you can see the alpha helices all coiled up from there. And then if you zoom in on the alpha helices, you can see the individual sequence of amino acids, over there. So that's just a way to like look at it big picture. Like if you zoom in on one polypeptide chain or subunit from quaternary structure, you can kind of see all the other structures within it. Does that make sense? Thank you. Okay, I'm going to talk about domains and subunits. I don't know how many of you feel the same way, but I felt like it was a tricky category for the first exam. So I thought I just put in, oh, you were saying you agree. You have a question? Oh, you were just saying you agreed. Okay, yeah, agreed. I thought this part was tricky to learn in this class, so I try to make extra slides, which I have linked at the end of this for you guys to view, just a special slideshow on domains and subunits. But I'm just gonna reiterate it again. If you guys remember, we used this analogy for for exam one review, but I wanted to just like re-explain it in a better way. So basically in this diagram, you have, these are just supposed to be representing people, but you're thinking of it in like the context of protein. So it's just kind of like an analogy thing. So I just want everyone to think of these as just people right now, and I'm gonna try to make the connection. So I want you to think of the person on the left as an individual, and then the person to the right of the people to the right of the arrow, excuse me, as a group, as two individuals who are part of a group. So you see like person A, person B, and they're like holding hands. So they're part of a group. So you have the individual on the left side and the group on the right side. And so on the group on the right, there's two people total in the scenario, although you could have more, but this example just has two people. And so this imagery is just the same thing as saying that you have two separate polypeptide chains, which make quaternary structure. So like on the right, you have the group that represents two people, but within the group, they're each their own individuals. So they're each individual people. And so if you have like one polypeptide chain that connects with another polypeptide chain, then you form quaternary structure. Because if you remember, quaternary structure is when you have two or more subunits. So you could say these individuals can be representative of like subunits, but when two people come together in a group, it's sort of similar as having two polypeptide chains that make up quaternary structure. And so basically when you have the two people in a group, each person is still their own individuals. Each person has their own hands, legs, heart, brain. They each have their own individual characteristics or properties. And these properties have separate functions. So person A has their own heart and it beats for them. It doesn't beat for like the whole group. So they have their own heart rate, and person B can have a different heart rate, but they have like separate functions. So that's kind of like saying how each polypeptide could have, in a quaternary structure, could have its own, each polypeptide in a quaternary structure could have its own domain. So things that have like separate functions. So an example I put in my analogy was just thinking of like, let's say this pink person was Tina, and maybe Tina was thinking about, and Tina was just thinking about like, I don't know, going downtown to get boba. So this could be Tina. And then this person, the blue person, could be Syed, and he's thinking of his kitten at home. And so his brain functions differently than Tina's brain. So they're like separate individuals with separate functions. And so since they both have individually functioning parts, you can say each person is just like similar in proteins, how each polypeptide has individually functioning parts with different functions. So that would be like having two different domains. And each polypeptide involved in quaternary structure can have domains with separate function. So that's just like, okay, sorry. And that's just like a like an analogy. I don't know if that helped or made it more confusing, but I try to make things like a real life application instead of like thinking of it in a science context to see if it would help. But did that make sense? Can I get a thumbs up? It made sense. Okay, most of you said it made sense. Great. Okay, and I included my little speech about the analogy there so you guys can look at that. So basically in 285, in this class, domains are sections of proteins that fold independently from the rest, and they have specific functions, like I said. Example here is we have ATP binding domain on this kinase here, and this kinase is just an example protein kinase A. I don't think we talked about it in this class, but it's just like a random kinase I found online, and it has an ATP binding domain, but it could also have different domains included in here too. And tertiary structure, if you remember, like tertiary structure can contain domains, but they do not have subunits. The term subunits is associated with quaternary structure. And so over here, subunits, I wrote here, proteins in quaternary structure can have subunits, which are the different polypeptide chains that make up the overall protein. And this example here, I don't know if any of you taken like kinesiology or many of classes that talk about like hemoglobin, but this is just showing hemoglobin and how it has like the 4 heme groups or just the 4 subunits, and each subunit is representative of a different color. And each subunit is a polypeptide. So there's like 4 polypeptide chains pictured here. And those make up an overall like mega protein, which is hemoglobin. But hemoglobin itself can also have domains within each of these subunits, or a subunit can have multiple domains. So that's just domains and subunits. Any questions? Okay, so since you guys don't have questions for me, I have questions for you. So we're gonna do like a true false thing. So you guys are gonna think about it and then we'll go through the answers. We'll see how this goes. Okay, first statement, true or false? A protein with quaternary structure could contain domains. True. Okay, great. Why is it true? No, no one want to explain why it's true. Okay, go ahead.
Made of polypeptide, and polypeptide contain such environment and difficulty, so they could have multiple domain.
Yeah, so I think you're right about that. So basically, as I said, domain specific region, one polypeptide serves specific function. And let's see. And yeah, so domain of one or more polypeptide chains of hemoglobin that help increase oxygen. That's just an example I had. But yeah, it's similar to what you said. A domain is just a specific region that serves a specific function, and you could have multiple embedded within quaternary structure. Does that make sense? Why that's true? Can I get a thumbs up? Thank you. Okay, great. Next question. True or false? One polypeptide chain can contain multiple independently folded functional domains. True. Okay, it's a pattern here. Okay, so yes, that's true. And that's just because domains fold independently, and it was just similar to what you said, and it just has its own specific function, which means that in one polypeptide chain or tertiary structure, you could find multiple domains in there. They could each have different roles within a protein. And that's similar to just what I said. Okay, another question. True or false? One protein or a single polypeptide that has tertiary structure will contain subunits. False. Thank you. Appreciate the participation there. Okay, false. Fake news. Why? Tertiary structure is defined as one polypeptide chain and the overall 3D shape that a protein folds into. Quaternary structure is when you have two or more polypeptide chains, and therefore these chains would be called subunits. Okay, one more question I have for you guys. It's a multiple choice question. Okay, three different proteins have the same domain in their structure. From this, we can infer that they A, have exactly the same primary structure, B, share at least one similar function, or C, cannot be three proteins from the same organism. Maybe that's a thinker. Maybe you guys take 2 minutes to think about it. Or does anybody have an answer? They want to raise their hand. It's like a more inspired question.
Yeah, I thought it's a very.
I tried to make these questions very Laura-like, you know, if you guys can tell. Yeah.
In a way that there's no answer. So
No. No. No, I made them Laura-like and so that it encourages you guys to think, critical thinking. Okay, anybody have an answer for me, please? B, very good. Okay. So if you said B, you were correct. Good job. Okay. Wait, should I explain? Okay, wait. I was gonna explain why. But yeah, so basically, domains are evolutionarily conserved. I don't know if you guys took evolution, but just for context, like domains that have roles in humans can have like the same role in frogs and whales and stuff. It's just like domains like serve a specific function. So like an ATP kinase in like a frog could do the same thing as ATP kinase in human. It had like the similar domain. So that's just like an idea about domain. They serve a specific role. It doesn't matter like where they are found. It will usually be the same function. Does that make sense? Can I get a thumbs up? Thank you. And this was just an example of polypeptide chain with many domains, which goes with one of the statements I said before. Okay.
So moving forward with our group interactions and bonds. So we have the amino acid structure, right? You have your amino group on the left side, your carboxyl on the right, which again which is important for your secondary structure. But then for your tertiary and quaternary, you have your R group, which is pretty much your side chain. And this is going to be what characterizes your amino acid. This is the only like part of the amino acid that's going to be unique to a specific one. And so because of that, that's going to determine its chemical properties and the kind of interactions it can make with other amino acids. Um, and so, for example, on the bottom, you have. Oh, and yeah, I almost forgot. So on the bottom, you have your hydrogen atoms, and they're important for your dehydration synthesis. And like I said, your hydrogen bonding in your secondary structure. Oh, did we move? So yeah, the hydrogen of the amino group and the carboxyl group, like I said. So moving forward, your first kind is going to be polar and non-polar. So pretty much, like we have on the left side, anything with a polar charge is going to be any group that has some electronegativity. So you're going to have one atom that's very electronegative, such as oxygen, right? It's going to attract a lot of electrons towards it. And you're going to have atoms that are low in electronegativity, so such as hydrogen, which are more likely to donate there. So because of that, like say in this water molecule, you're going to have a strong charge on one side and a weaker on the other. And so these polar are going to form interactions with themselves because you're going to have a positive end and a negative end. And on the opposite side of that, you have your non-polar interactions. These are between groups that have very similar electronegativity. So there's no preference for the electron to go to one atom versus the other. And typically you're going to see this between bonds of the same atom or between atoms of similar electronegativity. So such as you can have a highly electronegative oxygen bonded to a hydrogen, but if you have two oxygens bonded together, they're going to cancel out and it's going to have low in electronegativity, non-polar. But typically you're going to see with non-polar, you're going to see a lot of hydrogen, a lot of, or not hydrogen, a lot of carbon or methyl groups. So if you see that, then that's a good sign that it's a non-polar group.
Yeah, I think this should sound familiar if you took like if you're in Gen Chem or Orgo, like you guys went over this stuff. But I always think it's a tricky part for people to understand, like especially like when it comes to electronegative atoms like oxygen and sulfur, that they can be considered to be nonpolar if two of them are paired together. But that's just because the charges cancel out. So we just thought we'd include that in the description.
Right. It's not that the atoms themselves have charges, it's more the electrons and where they're going.
Electrons, yeah.
So moving on, then we have the different types of R group interactions, right? So you have the main ones, you have your non-polar, you have your charge positive, charge negative, and then your polar. And pretty much you don't need to know obviously like exact R groups, but just know the patterns that you see. So typically with your non-polar, like I said before, you're going to see a lot of carbon, a lot of methyl, which is just carbon bonded to 3 hydrogens. And you're going to see a lot of benzene rings, which is obviously just 6 carbons bonded together. Versus on your polar amino acid, you're going to see a lot of oxygen, a lot of nitrogen, right? Like we said earlier, that's what's going to make your hydrogen bonding or your partial charges. And then you also have positively charged, which is easy to tell because you can see the charge on the amino, or your negative charge, which you can also see a negative sign on the oxygen. And so pretty much your polar R groups are going to be making ionic bonds because of their electrostatic charges. And then your polar uncharged on the bottom are going to be making hydrogen bonds, like I said. You're going to have that oxygen interacting with the hydrogen or that amino giving up the hydrogen. And again, that's because of their difference in electron negativity. And then lastly, you have your nonpolar, and these are just going to be hydrophobic. So again, a lot of carbon bonded to carbon or carbon hydrogen. And these tend to be the weakest of the interactions, your van der Waals interactions or London dispersion. And again, because there's similar electronegativity or no preference for the electron to go one way or the other, that's going to determine its nonpolar. And again, nonpolar are going to want to interact with nonpolar, and the charges are going to interact with the opposite charge, and polar is going to interact with polar. So, any questions? And then again, like we said before, these R group interactions are what are going to determine the tertiary and quaternary structure. So again, based on whether you have a polar and a polar or a non-polar, whatever have you, that's what's going to form the actual like folds that then determine the property of your protein or the domains and such. So yeah, this is just a summary of the previous slide. Again, you have your ionic bonds, hydrogen bonds and your van der Waals or London dispersion interactions. That's good. Oh, we do.
Yeah, where's the hydrogen bond in the second,
In the secondary structure.
secondary?
Right.
Between
it's-
backbone atoms.
Yeah, it's between your back. It's between your carboxylic and your amino. So the oxygen on the carboxylic is going to attract the hydrogen from the amino, and because it's on the backbone, determining on how they interact, that's going to determine whether it's a alpha helix or a beta plated sheet. Does that answer?
Yeah.
So, and then we have the last one, which are your disulfide bonds or disulfide bridges. And these ones are simple because they can only be formed by the cysteine amino acid with itself. And these are strong covalent bonds, so they're used for stabilizing important tertiary quaternary structures. And again, with the whole concept of stability, not necessarily having a stable bond is better than having a weak one. It all depends on what the protein needs to do. So if a protein needs to be versatile, if it needs to be able to perform a lot of functions or not, or be able to be degraded, then you're not going to see these disulfide bridges. But if the opposite is true, then you're going to see them because again, they form that rigid, strong bond and add stability for the protein. Okay. And again, here's a summary of the different types of bonds. The important thing is to know which type of bond is in which level and what kind of groups make that. So again, your primary structure, your sequence of amino acids, those are going to be only covalent because again, that's the base of your entire protein. And they're going to be made, and those are going to be peptide bonds between the carboxylic group and the amino group. And then you have your secondary structure. These are going to be non-covalent hydrogen bonds between the hydrogen of the amino and oxygen of the carboxylic. And then you have your tertiary quaternary. These can be either covalent, non-covalent. Again, the non-covalent is going to be the ionic hydrogen and van der Waals interactions. And then the easy one, the only covalent one is going to be your disulfide. Okay. And then lastly, how does a excuse me? Lastly, how does a protein fold in 3D space? Pretty much two things. You're going to have the hydrophobic effect. So assuming that you are in a polar solution like water or blood or say, so have you, all your non-polar groups are going to want to cluster towards the interior. So they're going to move away from whatever polar solution you're in. And then on the opposite, your hydrophilic are going to orient themselves outward. And that's only so you can have interactions with for the outside solvent and the interactions between the interior core of the protein. Is that clear?
I think this next section is kind of short, so I'll go by a little faster. Okay, so there's a bunch of different ways to model proteins. The ones that we talked about were specifically 4 different types, and these are like the main 4 that you'll see overall. And we'll just talk about kind of like the advantages and disadvantages because sometimes Laura might ask about a certain scenario and then ask you what kind of modeling would be best suited for the scenario. So starting with ball and stick, or also known as wireframe, it is exactly like it says. You see your balls, which act as the atoms, and then you see the stick, which acts as the bond. And so the pro of this model is that it shows every atom and every bond in the protein, including the backbone and the R groups. So you can definitely see a lot of the details. However, a con is that even though every atom is modeled, it's hard to see the difference between backbone and the R group. So because you see every atom, every bond, and you see all of the details, it might get a little cluttered. And so you won't be able to see, it's harder to see the backbone, what part of it is the backbone and what part of it is the R group. And the wireframe is a little bit different. You don't see like the distinct atoms versus the distinct bonds. You'll see just the bonds. So it'll literally just look like lines. Oh, sorry, what's their question?
No, I was just doing the stick because
Oh.
when I think wireframe, I just draw out like this.
Yeah,
the
and then the
stick
next
on
slide
it.
is gonna have like a picture example of each one too. So then is the space fill model. So this shows also every atom, but more specifically, it doesn't show the bonds between the atoms. Instead, it shows just the atoms. And so it shows the space that it takes up. So it looks most closely to how it should look like in real life, or like in your body. And so the pro is that, of course, the overall shape of the protein you can see, and then especially the surface shape. So you can kind of see like the different R group interactions. Usually they're like color coded in textbooks and things like that. However, because it doesn't show you the bonds, and then it just kind of looks like a big bulb sometimes. You can't see the backbone of the protein, so you also can't really see the difference between the backbone and the R groups. There's also the ribbon model. This is mostly useful for when you're trying to look at your secondary structures. So it really shows you the alpha helices and the beta sheets. It's called a ribbon model because especially for the beta sheets, they look like waves of ribbons. So that's easy to remember. But unfortunately, the con for this is that you can't see the R groups. They really focus on the secondary structure for this model. And then lastly, you have the backbone model. This shows only the path between the polypeptide backbone. So it also does not show the R groups, pretty similar to the wireframe model. So yeah, I think wireframe might actually also be called just the backbone.
Yeah,
But
typo there.
little typo, we'll fix it
we
before
will
we send
fix
it
it.
out. Yeah.
Yeah.
Okay. And then also remember, so you can have, it's not just one model that you have to decide on. Sometimes there might be a best version, but models can also overlay on top of each other. So oftentimes you'll see like ribbon models to show the different secondary structures, but you'll also occasionally see like maybe whoever the researcher is or whoever's like publishing decides that they also want to show the different R group bonds.
Yeah, I was gonna say models can be like layered on top of each other sometimes, and I think she showed an image of that in her class slides. Though I think for the most part for these questions, you just have to choose the best model for a situation, like a model, like one model for a situation.
Yeah, it's just good to keep in mind that it is a possibility. So you might not have like surprises if she,
Yeah, usually she just says like, what is the best model?
yeah.
And then you choose.
Yeah, and we'll have an
depending on-
example.
We have a
question
good
too.
example question.
Michelle.
Yes.
Is bond stick and wireframe, are those two different things?
Yeah, so a little typo. So wireframe is like backbone. That's like the skeleton. Ball and stick, you can see both the atoms and the bonds. Whereas
Yeah.
wireframe, you see
The
just the
circles
backbone.
and like the
Yeah.
like sticks they're drawn
Yeah.
to.
For
That's
if you
ball and
ever
stick.
take like, if you ever end up taking Orgo for anyone, like the model kits that they ask you to use, that's a ball and stick model.
There you go.
Yes.
Another-
I used that on my exam. I remember that kid.
Yeah.
That's what it
Yes.
is.
So on exam 1, I remember the question was asked like about the model.
Mm-hmm.
But it was
Oh,
Well,
oh.
I think it's blended.
No, it's okay. Okay. So
is that gonna be? I know you guys.
If it
I
happens, yeah.
haven't seen her exam, but I do remember that picture you're talking about. And the exam was printed in black and white. And I said, Laura, they cannot see the model. I said, can I please project it on the board in color, in vibrance? And she said yes.
Also,
So
if that ever happens, like don't be afraid. Like
Don't
most of
be
us are gonna
afraid to speak
proctoring.
up.
Yeah.
because even me, people raised their hands and said, can you please help me with this question? I said, I can't see. I don't know what's what in this question. So she was able to project it in color. So for things of that nature, please speak up about that kind of thing. She's not evil. We're not evil either. We want you to do well. We're not gonna, be like, no, we can't. But I think, and I think she tries hard to make the final not something that you would like have to struggle to see an image or something. I would hope that she'd make it clear. But if that was the case, she would try her best to like either describe the image to you or have some sort of alternative projection. But I'm not sure if Topman Gym has the projection thing, but I'm hoping that like that will not occur. It'll be simple to be able to tell. And we have an example question coming up next where like I feel like if you think it through, you'll be able to tell which model is the best to apply for the situation. So does that make sense?
Mhm.
Good question.
It shows you like the,
Yeah, it
you'll see in the
does.
this next picture, yeah, they're like color coded
They're color
usually.
coded, but it's a whole mess space fill.
Yeah,
Like it's just you just see everything
big
all
one,
at
yeah.
once, and because it looks the way it shows like the surface area that the protein takes up in real life. So there's just a lot going on, and it's really hard to discern specifically like the backbone of the protein. You'll see that these models like they're good and they have pros and cons. Like each one showcases a specific thing
Yeah.
that another one doesn't.
So like, for example, space filling, because it shows you like the amount of space that it takes up, and you would want to use space filling if the question is something along the lines of like, oh, I want to know like how it looks like in real life, or like how
Two
much space it
proteins
actually takes up
interacting.
to, yeah.
A protein and a ligand interacting, and maybe you wanted to study the surface interactions, like which one would best show you what was going on. Like you could see on the surface of space fill, like what it takes up. I think that would be like maybe an example of that. But we do have a very good question that the TAs made for you guys, especially to answer about this stuff.
So these are just the different to visualize what it is. So this is the ball and stick model. This is the space fill model, backbone or wireframe model, and then the ribbon model. So again, like they look like strings of ribbon.
Yeah.
Any questions about like how they look like, or how to like figure out which one is which? Cool.
Question time.
Okay. Michelle made this question, not I'm not like self-centered, I promise.
Yeah, I made a question.
So a scientist named Tina Chen is trying to do a presentation on prions disorder called mad cow disease. Mad cow disease is caused by a severe misfolding of prions proteins in humans, which can cause them to take on a weird shape. In particular, these evil prions can have large differences in secondary structure compared to normal prions. Disease-causing prions can be recognized by their clumps or thick stacks of beta sheets. Tina, unfortunately, did not pay attention in her Bio 285 class and is unsure of what model to use. Can you help her?
Hey, you change it. I said Tina's a very bright scientist. Can
Well,
you
I
help her choose?
I didn't want them to think I was like self-centered.
Okay,
So anyone
Ooh.
has it?
that was
Is that,
fast.
is that a consensus?
Do we
Yeah.
all feel like she's correct? Anybody want to rebuttal? No?
All right. Well, you are correct. It is the ribbon model. The way that, do you want to explain like how
Why?
you know, or-
Oh, because it says that they have large differences in secondary structure. Like, beyond so automatically. See with these like base sheets, and then it says they can be recognized by their clumps of big stacks of beta sheets. So then you know it's ribbon model.
You solved it so
Exactly.
quick. Part of me thinks I should have made it hard.
Yeah. and
But
if
you did a good
can't
job.
s- if you can't
Correct.
see, I think it says.
We put a joke in there.
Yeah.
Or I did. We got the cows here and they said, and you were worried about mad cow disease, and they got the coronavirus paper they're reading. Funny. Anyways. Oh, no, wait.
Wait.
Real quick, just wanted to say, this is not a made up scenario, in case you're curious. Mad cow disease is a real thing.
Mhm.
When I took the class 2 years ago, she actually taught us about prions and how you have good prions in your body, and if they come into contact with like bad prions, they can misfold and aggregate, and you get a lot of beta sheets
Yeah,
that
exactly.
are clumped up there.
So
And
that's why you
actually-
want like the ribbon model too, because for instance, if you use the ribbon model, this is the only way that you would be able to tell whether they're normal or not, because on disease causing ones, you'll see a lot of these
beta sheets. Yeah, and also just be careful of what meat you eat, because that's what causes it. I think eating like different whatever. Anyways, that was not an advertisement to be vegan or anything. That's just, side note. Anyway, just be careful.
So next we're gonna go into dissociation constant and ligands. I know, I think during this section, some people were confused about this as well.
This is hard stuff.
Yeah.
I never got that stuff.
All right, so starting off with protein ligand interactions. A ligand is anything that a protein binds to. So sometimes they're also known as substrates. So for example, here you have your protein, and then you have CAMP. CAMP acts as the ligand or the substrate that's binding to the protein. The interactions don't always involve a binding pocket. So this example shows a little pocket that it kind of fits into, but it can also be like a flat surface that it's just kind of interacting with. It doesn't always have to be a nice little like puzzle piece. And also the proteins always have to be oriented correctly, as well as the amino acids. So orientation is definitely very important because depending on how your ligand and protein is oriented, that determines the different R group interactions that can happen. So say for instance, if you flipped camp, like it went in where like now this is on this side and then these R groups are on this side, you probably wouldn't have that good of a binding because it's not going to have the right interactions that can occur.
Or if you swapped out one of these amino acids for a different one.
Yeah.
Ooh, a good question. Think
That's-
about that.
She really likes doing those type of questions.
If you swapped out one for another of a different chemical group,
Mhm.
like what would happen?
Yeah,
What would the
so
interactions?
this is definitely where like what Saya was just talking about with the R group interactions, understanding your R group interactions, understanding all the different types that can happen would really help with this kind of question. Because for instance, if arginine was replaced with, say, a
Leucine,
like a
something
leucine
non-polar.
that becomes negative, well, now you might not have this interaction because they're going to be repelling each other.
Yeah, sometimes if you change one amino acid for something else, it could result in maybe like a better interaction or no interaction at all. Or if you swapped it out for something of the same amino acid category, there could be like no change. So it just depends on the situations.
Did you have a question?
Yep.
Does charge or size affect it?
More?
I don't know. She always used to say size and chemistry.
Yeah, I would say
Focus more on size and
she
chemistry.
usually wouldn't actually like has she asked the question before like that? Is that what you're asking? Or that's it. Yeah, I think she does usually group together size and chemistry.
I think charge. I don't remember.
Well, charge would be like chemistry.
But yeah, plus or minus charge, like a positive, polar positively charged amino
Yeah.
acid being traded out for like polar negatively charged, like you could make implications as to the
Mhm.
changes on that. But I remember she always used to say like when you writing about like what would happen or having a conversation about like with somebody else about like what would happen, explaining the size and chemistry. Because like the specific affinity for something like it has to be the correct size and chemistry. And if you exchange a small amino acid for a larger one, there could be implications or like chemistry, like size and chemistry together have like a big impact on the interactions.
So usually the type of question you would want, usually it's like a free response question, right? So you would want to make sure, unless she specifies, that you explain both if the size affects it and if the chemistry affects it. Like you don't want to just talk about size and you don't want to just talk about chemistry.
yeah.
Does that make sense? Good. Awesome. Yeah, okay, cool. So then you touched on my last point. Perfect. All right, so then going into affinity, affinity is the strength of the interaction between the protein and the ligand or whatever it's binding to. So strong affinity means that they fit together really well. Weak affinity means that they're, not a perfect match, but that doesn't necessarily mean that's good or bad. Definitely keep that in mind. So affinity depends on two main things, the number of bonds between the proteins and then also the strength of the bonds. So the more bonds you have, the stronger the binding because it's connected more, and then the strength of the bond as well. So generally, from weakest to strongest, it would be van der Waals or like those electrostatic interactions, and then hydrogen bonds, and then ionic bonds, and then for this class, covalent bonds are the strongest. I know we usually address it in class as well, but like in chemistry versus like bio, it's a little different. But for the sake of this class, covalent bonds are the strongest. And then you want to also consider the type of bond and the angle of the bond. So that just be like angle, like as I said before, the better the fit, the stronger the affinity. So if you're angled weirdly, or like if the ligand is not going in at the right angle, then it might not have like it might not be close enough to the protein to have the proper interactions that it needs, things like that.
Yeah, not the degrees, not like a certain degrees amount or
Yeah.
anything, just orientation.
Yeah, just like for instance, like if the ligand needed to put this with over here, and then it's like angled a little oddly where it turned a bit, then like it ends up over here, it might not have as good of affinity as it could have had because it's not close enough to where it had to be. Does that make sense? Cool. Yeah. All right. So then dissociation constant, also known as KD. This is like the formula for dissociation constant, usually it's less about the formula and more about the concept of dissociation constant that Laura focuses on. But for the sake of understanding each part, P stands for protein, but more specifically, the concentration of free protein that's available. This is the concentration of ligands available. And then this is the concentration of complex. C is for complex. So that just means proteins are binded to ligands, or ligands are binded to proteins, same thing. And it's a way to measure and describe the protein ligand interactions. The dissociation constant is an average rate of measurement of how often the proteins dissociate. So that just means like how often or how likely are they to separate from each other. So the lower your dissociation constant, the higher your affinity, because that means you are unlikely to separate. And if you're unlikely to separate, that means they're really stuck together. So you have a high affinity. Does that make sense? Cool. Yeah, so they are inversely proportional. So the lower the KD, the higher the affinity, vice versa. Higher the KD, lower the affinity. Yeah. Oh, and then something that when I took the class was always confusing for me, she would talk about like how the concentration of proteins equals the concentration of complexes. So that just means at equilibrium, your concentration of ligand is equal to your KD. And that only happens at equilibrium when half of the binding sites are filled. So in other words, if you have 100 proteins and you have 50 free proteins, then the other half, which is also 50, are filled. So that's why you have 50 free protein and then you have 50 complexes, and then that would be equal to each other. And then that's at like perfect equilibrium. So your concentration of ligand equals your dissociation constant, because if they're equal to each other, that makes it 1. So 1 over 1. So they like cancel out. So mathematically, KD equals L. So that's like the math of it. I know some people are like wired that way, if that helps a little more. Yeah. Wait, was that a question?
No,
Or was that clear?
no,
Oh,
it's
okay.
good, no, good.
Sorry. All right, awesome. Was there any questions before we move on?
No questions? Okay, we'll continue. Me again. Sometimes I feel like I sound better on 1.5 speed, or is that just a me thing? Good thing you guys have YouTube for that. Anyways, protein trafficking. Okay, so protein trafficking. Basically, all proteins start their life in the cytosol. That's where they're born, I guess. All proteins are made in the cytosol by a ribosome. And there are 2 types of ribosomes that we talked about. We have the freely floating ribosomes, which are just regular ribosomes. And then you have ribosomes that are bound to the ER membrane. These ribosomes specifically are attached to ER so that proteins going into ER just go right in the translocation channel. Proteins that are heading to the endomembrane system are made by ribosomes that latch onto the ER membrane. I just said that. Anyways, and then proteins that don't go to the endomembrane system will be made by freely floating ribosomes that are just floating around in the cytoplasm. So those types of proteins are cytosolic proteins, obviously, mitochondrial proteins, nuclear proteins, and chloroplast. I don't think she talks much about that, so just ignore that one. Anyways, does this make sense? Thumbs up. I rely on the approval, so I know to move forward. Thank you. Anyways, so this is just a visual representation of what I just said. You have your freely floating ribosomes and then membrane-bound ribosomes. Any questions there? Okay, endomembrane system. What does that mean? So endomembrane system is just a bunch of membrane-bound organelles that a protein can travel through if a protein is going through the system. And they all have membranes, and examples of these are ER, Golgi, lysosome, and the cell membrane itself also is part of it if you had a protein being secreted out of the cell. And another way to describe endomembrane system is just a secretory pathway, because proteins will travel from each of these membrane-bound organelles and travel across vesicles. I have the image of that in the next slide, but that's just the interesting thing to know. And every protein that goes through the endomembrane system starts in the ER. So as you can see, if a protein needs to end up in a lysosome, it has to go through the ER first and go through this endomembrane system and Golgi bodies, and even to get outside of the cell. And by default, if a protein only has an ER localization sequence and nothing else, it will continue to travel through the endomembrane system until it gets out of the cell. And a lot of people, I think, got confused by this one on exam 1, or just like when we learned the material. But this is basically because if a protein is going to the ER, or it lives and works in the ER, it's an ER protein, it's supposed to be there. That protein is supposed to have 2 sequences. So we'll have the ER import sequence and what we call ER retention sequence. And if you remember from the mechanism of ER transport, the ER import sequence is N-terminal, and once the protein is in the ER, it gets cleaved. So then the rest of the sequence is read, and you find the ER retention sequence in the C-terminus. So that's what allows the protein to stay in ER. But if the ER import sequence gets cleaved and it had no other sequence, it would just continue through the endomembrane system till it gets secreted out of the cell. Does that make sense? Perfect. Okay, and a diagram so that you guys can see visually what's going on. But same thing as I said, all these membrane-bound organelles, lysosome, ER, Golgi, etc., and its secretory vesicles, they're all part of endomembrane system. And even proteins that make it outside of the cell have to travel through this special pathway. Okay, practice question, hooray. I don't know why they all say hooray, anyways. So plutonium is a protein secreted out of alien pancreatic cells and travels through their bloodstream. Given what you know about the mechanisms of protein sorting and trafficking, what types of signal sequences do you think that protein plutonium has? Anyone? Okay, I'll give it some time. It'll come. Someone else, sorry. I just want to give people a chance to like throw in ideas. Anyone else? They make it too hard. Anyone? Bueller? Bueller?
Is the ER localization the one that brings it to the ER in the first place, or is the one that makes it stay?
Okay. Is there anyone that like understood this question and can answer it for me first before I answer that? Okay, go ahead.
These ER localization first,
Yes, so that's correct.
and that's it, I guess.
That's correct. Why? So you said it needs ER import only.
Once it has that localization, if it doesn't have retention, it's just gonna go through all the way out the cell.
Okay, that is correct. Thank you. So we're gonna just go through this. So because this special protein is secreted outside of the cell, it must enter the endomembrane system. How does it do that? You need the ER import signal sequence. So that's what you need to get it in the endomembrane system. Because remember, I said for any protein that needs to go somewhere in the endomembrane system, whether it's lysosome, out of the cell, etc., needs to start with the ER. And so you have the ER import sequence. And if you recall, ER import sequence is N-terminal. We're going to talk more about the ER import, the mechanism, but this sequence gets cleaved off once the protein enters the ER. And when this happens, the sequence is cleaved, so then the protein has no other sequence. Once it gets in the ER, it's not going to be able to stay there, right? It will just keep traveling through the endomembrane system until it reaches the cell membrane and leaves the cell. So this is just basically what I wrote here. Does that make sense now for people who are still like thinking about it? Why the answer is just ER import sequence? Why it wouldn't have any other sequence? Can I get a thumbs up? Okay, good. Almost all of you gave me a thumbs up, which means I was like, okay, it was a fair question. I thought maybe it might be too tricky. Okay, so any of you had Professor Phillips from 151? You remember this diagram? Anyways, he had it in this class. That was my professor, great guy. Anyways, so this is just a diagram that you can use, you know, when you're studying, you can figure out where a protein is gonna go. ER signal sequence, yes. Then it continues. Ignore this anchor sequence stuff, doesn't apply to this class. But protein travels through the ER lumen. This is like the whole endomembrane system thing. Golgi body, does it have another signal? No. Gets secreted out of the cell. So that's how you would like follow the sequence for the question I asked before. And you can just work your way through stuff. If there's no ER signal sequence, gets translated in the cytosol by a free ribosome, etc. You can figure out where stuff goes. Just thought it was cool to include that. Okay, next up, signal sequence is basically a stretch of amino acids embedded in the primary structure of a protein. What does that mean? It just means that the signal sequence is already incorporated into an amino acid sequence. So it's not something where a protein is translated and then we tack on a signal sequence at the end. No, that's not true. Signal sequences are always part of the protein itself. It's never added after the fact. As an analogy, I tend to tell people to think of a signal sequence as a unique address. Tells a protein where its destination will be, kind of like a zip code. When you're mailing a package or something, you put the zip code for Amherst so they know to send it here and not somewhere else. Okay, so basically every time a protein goes to the ER, it has to have an ER signal sequence, which is just another way of saying ER import sequence. And the reason why that is because ER signal sequence is recognized by SRP, which will bind to a newly made protein and take it to the ER. And it binds to the protein in the N terminus and it takes it to the ER. And this is important because if a protein is missing an ER import sequence, SRP won't recognize it and it's not going to be able to bind and take it to its destination. So if a protein, so this is why if a protein does not have any sequence at all, then it's never going to leave the cytosol because all proteins, like I said, start their life in the cytosol. So if she tries to trick you and ask you a question where you notice that there is no sequences at all, or if she had like a protein that had sequences and she removed them all and there was no sequence, where would the protein end up? It would just stay in the cytosol. So yeah, and any protein, like I said, that needs to leave the cell or be secreted will go to the endomembrane system. So that's just that stuff. Any questions? A thumbs up if it made sense. Okay, great. More stuff on signal sequences. So signal sequences take proteins to their final destination. So in some ways, signal sequences can be considered to be binding sites for other proteins whose purpose is to aid in the transport of a protein to its location. An example of this, which we looked at way back at the beginning of the semester, was Important, which is pictured here. Important will recognize the NLS on a cargo protein or a protein that's intended to go to the nucleus and will bind to that special sequence on the exterior surface of that protein and bring it into the nucleus. Same thing with SRP. It recognizes the N-terminus sequence on the ER protein and brings it to the ER. So this is why disrupting signal sequences can be harmful to a protein because it's not going to be able to get to its destination, and maybe it's not going to be able to perform like the same functions. So yeah, and so for nuclear import, proteins that enter the nucleus are fully folded and fully made. This was like a phrase Laura used when I took the class, but it essentially means that the protein is made in the cytosol because it's a nuclear protein, and then it just gets taken into the organelle as it is. In other words, there's no deconstruction and it doesn't enter through the nucleus like spaghetti, as we see in some of the other structures. I always say like spaghetti because it unfolds for the ER import. But yeah, so for nuclear import, protein will enter exactly as its normal 3D shape is. And why is this the case? Because nuclear proteins can enter the nucleus because the nucleus has pores that are big enough to be able to fit proteins going in there. And this process is highly regulated. So only proteins that are supposed to go in the nucleus will pass through this special nuclear pore complex. And yeah, so that's pretty much summary of what's going on in this slide. Exportin has a similar process to how it's able to bring in cargo proteins, how it's able to take cargo proteins out of the nucleus, but it's done using a different sequence than Important called NES, or nuclear export sequence. Does anybody have any questions on what I just said, or does that make sense? Oh, question.
Are these, like the NLS and NES, are these sequences being chopped off?
No, big difference. So for nuclear proteins, the signal sequence is actually embedded on the exterior surface of the protein itself. So it's just embedded on like the front of the protein or the surface. So the signal sequence is not cleaved. It's part of the entire protein. You can't really cut it out. It's just part of that protein. It never gets cleaved for nuclear proteins. So that's actually a good point to bring up.
If a protein has both NLS and NES in the two cycle?
Not really sure about that, but proteins that need to work in the nucleus, they'll have NLS, which gets them from the cytosol to the nucleus. But I remember on the exam one, she asked a question that if you had a protein that works in a nucleus, whether you programmed it to be able to go to the cytosol, what if you could add a sequence to it? So you could add, maybe hypothetically speaking, an NES to a protein that already has an NLS, so that it's able to do that.
Yeah, theoretically, it would be able to do like just multifunction.
Multifunction.
We don't know if like.
We
in
don't know
real
if it
life
will
there is
cycle
this,
through,
yeah,
but it's a possibility that it'll be able to perform two functions.
it could do it.
So it could do it. For example, importin, right? Importin works in the nucleus during nuclear import. You'll see when we talk about nuclear import, it just like stays, it like works in a nucleus. But then like it has to get back out to like the cytosol somehow. And so like I think it has the help of other proteins. I think I'm like, once I get there, I'm gonna talk about that. But that's actually a good question because she did ask it on the exam. I don't want to like, I think I'm confusing my brain a little trying to think about it unless I have this slide in front of me, but I'm gonna get to that stuff. Okay. Any other questions? Really quick, I just gonna show short video on ER import. I think it was a hard part of understanding this thing, but it's like 2 minutes. Thank you.
All protein synthesis in eukaryotes begins in the cytosol, where ribosomal subunits come together and begin translating an mRNA near its 5' end. Soon after the protein starts to emerge from the ribosome, it may become directed to the endoplasmic reticulum, or ER. It is targeted to the ER if it is ultimately destined for incorporation into the ER, the Golgi apparatus, lysosomes, or plasma membrane, or if it is destined for secretion. In mammalian cells, most proteins enter the ER co-translationally. These proteins have an amino acid sequence called a signal sequence, which is typically located at the amino terminus of the polypeptide chain. Signal sequences span about 20 amino acids and include a stretch of hydrophobic amino acids preceded by a basic amino acid. As a signal sequence emerges from the ribosome, it is recognized and bound by a signal recognition particle, or SRP, consisting of 6 polypeptides and a small cytoplasmic RNA. The SRP binds the ribosome as well as the signal sequence, and in so doing, stalls translation. The SRP targets the entire complex to the rough endoplasmic reticulum by binding to an SRP receptor on the membrane of the ER. GTP molecules bind to the SRP and the SRP receptor. The GTP binding triggers the transfer of the signal sequence from the SRP to a membrane channel called a translocon. Hydrolysis of GTP to GDP leads to the dissociation of SRP from both the receptor and the ribosome mRNA complex. Transfer of the ribosome mRNA complex to the translocon allows the signal sequence to interact with short hydrophobic side chains located in the narrow neck of the translocon channel. This interaction opens the translocon by moving the plug away from the translocon channel. As translocation proceeds, the signal sequence is cleaved by signal peptidase, and the polypeptide is released into the lumen of the ER. Thus, the process of protein synthesis directly drives the transfer of growing polypeptide chains through the translocon and into the ER.
Okay. This video had some parts of it that's like not from 285 specifically, but I don't want to confuse anybody. So just please focus on the ideas mentioned in 285. So the whole GTP SRP part and the plug, like don't focus on those small things. Question?
I thought ribosome was tied to the ER.
Yeah, it is. I think the video like has a mistake in that part of it, but yeah, so sorry. So the membrane bound ribosomes are the ones that bring like translate the proteins that go in the ER. Good point. Which is why the video is just for like fundamental understanding, but I thought I'd show it because somebody, when I was helping somebody with the in office hours, they said it was like helpful. So I just thought I'd show it. But an important part of the video I think you guys should like pay attention to is the co-translational part, because as soon as you have the N-terminus sequence of just a few amino acids, like they said, 6 peptide sequence, like as soon as you have a protein that's barely even completely created, so just like a baby amino acid sequence like this, it's immediately recognized by SRP, which binds to the N-terminus and quickly brings that protein into the ER, where the protein continues to be translated and pulled through the ER. which is why the ER import happens much faster than some of the other imports. So if you ever get a question where you have to decide between, oh, a protein has this type of import and the other kind, and you don't know where it's going to go, just keep in mind the ER import is the fastest process because it's like of the timing. You only have like 6 amino acids that are produced in the N-terminus, but that's immediately recognized by SRP, which is already bringing it into the ER. And by the time the protein is like completely made, it's already in the ER, it's finished, and the signal, the N-terminus sequence gets cleaved and it's already in the ER by then. So if there's any other sequence in there, it's not going to be able to go anywhere else because it would already be in ER by the time it's like fully created. Does that make sense? Thumbs up if that makes, thank you. And then the mitochondria was tricky for people, so really sorry, but another video. It's 1 minute though, so I'm just gonna play it.
Proteins made in the cytosol must therefore be sorted and selectively delivered to their proper destinations. Precursor proteins destined for a mitochondrion have a short segment of amino acids, the signal sequence, that targets the proteins to this organelle. The signal sequence has affinity for a receptor on the mitochondrion surface and delivers the precursor protein to a translocation apparatus for import. At a contact site where the mitochondrion's 2 membranes are close together, the precursor protein snakes in an unfolded state through 2 sequential protein translocators, one in each of the mitochondrial membranes. Inside the mitochondrion, chaperone proteins are required to help pull the protein in. Chaperone proteins bind to the precursor protein as it appears on the inside of the mitochondrion, and thereby prevent the protein chain from backsliding through the translocation tunnel. Once inside, an enzyme called a signal peptidase cleaves the signal sequence, which is no longer needed, from the precursor. The chaperone proteins are released as the protein chain folds into its 3-dimensional structure.
Okay, so basically, I just want people to note their similarities and differences between ER import and mitochondria import. The mitochondrial protein will only get carried over to the destination of mitochondria once the protein is in the half-folded state. So that's just a difference between the two.
Proteins made in the cytosol.
Okay. Okay, so now I'm going to talk really quick about mitochondria because I think this was like a point of confusion from when we looked at this stuff from that week. So any protein that goes in the mitochondria doesn't come back out, and these proteins are carried by free ribosomes in the cytosol, which is very different than the ER import, right? And so they will be taken over to the mitochondria once a polypeptide is in the half-folded state, which is different than ER because obviously the ER, the protein is just like a small baby sequence of just a few amino acids, and then it gets taken in that process. But basically at the mitochondrial membrane, the protein will bind the signal sequence and draw it into the translocane channel, as you saw in the video. And like I said, protein will only enter the organelle in a half-folded state and in an unraveled form like spaghetti. With the ER import, it also enters unfolded like spaghetti. Those are like similarities between the two. And so once it makes its way inside the mitochondria, signal gets cleaved because the signal is N-terminus, I think, as well. So the signal gets cleaved, and special chaperone proteins help refold into the correct orientation. Does that make sense for this one? Okay, and now this is a summary slide with just all your different organelles. She, I think, focuses on these main 3. Let me know if there are any more that you've seen, but I think she only focuses on these main 3: nuclear, proteins, ER, and mitochondria. And so as you can see, as you were bringing up earlier, with nuclear protein, signal sequence stays on at all times. It's never cleaved. And with the ER import, mitochondrial import, the signal sequences, once they enter the organelle, are cleaved. So those are big differences there, big distinction. With the nuclear proteins, the signal sequence, is part of the folded protein. So in the like tertiary structure, so it appears on the protein surface. Whereas with ER import and mitochondria import, with these proteins, your signal sequence is embedded into its primary structure, so within the sequence of amino acids, and it's towards the beginning of the sequence in the primary structure. And with nuclear proteins, you see that proteins enter fully folded and fully made. They enter in the shape that they're supposed to be, the correct shape. Whereas ER and mitochondria, the proteins enter unraveled kind of like spaghetti and they get refolded once they make their way into the organelle. And yeah, this is just a summary of everything that I spoke about. Any questions? No? Okay, I put a practice question. You might recognize this question from exam 1, but I just figured I'd put the signal race question. So polypeptide has both the mitochondria sequence and ER sequence. Where is it going to end up? Anyone? Go ahead. Yeah, I'd say so, but I'd say there's an answer that's more correct based on the timing of things, of the events. So can anyone give me an answer as to what they think it would be? Yes, in the back? ER. Okay, great. Why? Yes, that's true. I was gonna help you elaborate, but I know on paper, like you'd elaborate and everything. But yes, that's the reason why. Just because ER sequences are N-terminus, and immediately as you have the little baby amino acid sequence, it immediately gets recognized by SRP, which will grab the amino acid sequence and pull it into the ER and takes the protein through the translocation channel. So I call that like a newborn protein, like a newborn amino acid sequence or a baby sequence. So at that time, there's a whole co-translational thing happening. So the protein is already being moved into the ER before the entire thing has been made. You only have a little piece of it, but it's being strung through the ER. And that's happening so quickly at that point that it wouldn't make its way to the mitochondria. And so like she said, thank you for your answer. The ER localization happens the fastest due to this co-translational mechanism. And translation happens at the same time when the protein's being localized. And yeah, so by the time the protein is fully made, it's already been pulled into the ER like long spaghetti. And then the N-terminus gets cleaved and it prevents the protein from leaving the ER. And yeah, and so the reason why it wouldn't, I don't think it would be mitochondria if you were to be asked something similar like this, is just because the mitochondrial proteins don't get trafficked until they reach the half folded state. So that is when you have like half of that polypeptide, and that takes a lot longer than the ER import because ER import is more of an immediate thing when you have like just a couple of amino acids and that gets recognized by SRP, and it's immediately taken to the ER. Can I get a thumbs up if that made sense? I'm happy pretty much all of you said it made sense because I remember this was on the exam and like everyone was asking me about it and the other TAs and it was a stumper back then. But now you understand why it makes sense, right? Okay. Oh, another practice question. Okay, so I gave a scenario in here. Let's say you have a 10 peptide sequence of hydrophobic amino acids that's required to bring a protein into the ER. If you mutate 6 of the 10 amino acids, do you think the protein would still be able to enter the ER? If we change the protein, let's say we had like, I don't know, let's say we had like 6, let's say we had a 10 peptide sequence of like leucines, and I changed 6 of those to become serine. Do you still think that this protein would be able to enter the ER? You shaking your head no. Would you like to tell us why? No? You don't want to tell me? Okay, fair enough. I'll just say it. The answer, no. Why? Okay, signal sequences, like I said, function as binding sites for other proteins. And if you change the signal sequence, you're tampering with another protein's ability to recognize specific binding site. So by changing the chemistry of the ER localization, ER import sequence, you're preventing it from being recognized by SRP. And yeah, so that's basically what's going on there. And if you mutate the signal sequence, then SRP might not be able to recognize that same sequence, and it's not going to bind to that protein that's supposed to go to the ER, and that protein could possibly be stuck in the cytosol. Yeah, so that's that question. And did that make sense, everybody? Can I get a thumbs up? Yeah, she asked me that years ago on like a problem set, and I was like, oh, okay. Anyways, nuclear import and export. I'm just gonna try to roll through this. So if you guys remember, we talked about GEPs and GAPs way back when this was like week 3 or something. But basically, GAP is what it stands for is GTPase activating protein, which could be confusing for a lot of people to understand at first because you're like, wouldn't that activate a protein? But GTPase, essentially, GTPase is an enzyme that hydrolyzes GTP into GDP. And so if you're if you're stimulating the GTPase activity of a protein, you're causing it to switch between the GTP and GDP nucleotides. And so basically, a GTPase on its own has the ability to hydrolyze GTP into GDP, but it does so at a very slow rate, which is why it enlists the help of the GAP protein that accelerates this GTP hydrolysis process. And so it's able to help a protein do that faster. You'll see that when we talk about RAN in nuclear import and export, because RAN is an enzyme, it can hydrolyze GTP into GDP because it's a GTPase, but it can't do it very fast, so it's slow. So GAP will bind to RAN and help it to accelerate its intrinsic, help accelerate RAN's intrinsic GTPase activity. Did that make sense? And so the end result is when you have GAP involved in binding to an enzyme or a GTPase, GAP will make a GTPase inactive. And the end result is that RAN would be bound to GDP in the case of nuclear import and export. And yeah, so that's just the role of GAP. And yeah, I'm gonna talk about GAP and then we can go and talk about, yeah, go ahead.
Someone said to me once for GAP, like a really nice way to remember is that it's like GAP, even though it's activating protein, so you would think that it's gonna activate it into like GTP, but instead you wanna think of it like it's literally causing a gap. So GTP to GDP, it's like making a gap between the two phosphates. So that's how you can like remember that's going from a triphosphate to a diphosphate.
That's a good way to think of it. And actually, you don't have to know exactly what GAP and GEF stands for. I just like to explain it so that like students can understand its role much easier. You'll see when we talk about GEF, but knowing what GEF stands for is going to help you distinguish between the two. But GEF stands for guanine exchange factor. Thank you. I should have known that. Anyways, so that's what GEF stands for. And GEFs, what they'll do, they kind of have an opposite effect than GAP, right? Because GEFs cause enzymes to become active or proteins to become active. So GEFs will bind to, in the case of nuclear import export, which we're going to get to, GEF will bind to RAN, that is an inactive form of RAN. And when RAN is inactive, it's bound to GDP. And when GEF binds to RAN, it causes a conformational shift, which is just a fancy way of saying shape change, such that RAN no longer has good affinity for GDP. And that's what happens when GEF binds to RAN. And so what happens, because this is all happening inside of a cell, inside of a cell, there's just more GTPs floating around, GTP molecules. So when GEF binds to RAN, it just encourages GTP to move into the binding pocket of RAN. So GDP diffuses out and GTP molecules will diffuse into the binding pocket. And when RAN is bound to GTP, it causes RAN to become active. But yeah, so GEF is just utilized in the processes of nuclear import and nuclear export. And I'm gonna talk more about what it does in those slides. Any questions? Okay. Something Laura always told me about nuclear import, which I forgot to say back when we were learning this stuff, is just think of nuclear import like a bus stop. So Important would need to take its passenger, the cargo protein, into the nucleus and then drop it off and then pick up more cargo proteins. So I just try to think of it as like a bus stop thing. So Important is the bus for the cargo protein, but then RAN is, I feel like, is the bus for Important to bring it out of the nucleus. Because Important does not have a nuclear export sequence, it just has an NLS. So Important cannot leave the nucleus by itself without the help of RAN GTP. So RAN GTP is kind of like Important's bus. But anyways, just gonna go through just what happens in this pathway. So Important will bind to the NLS on the cargo protein, the protein that is destined to go into the nucleus. So then these two will enter the nucleus. And then the next part is just that you have the GEF, which I don't know why they don't just like draw out the protein. You just have the word GEF, so it's there. GEF will bind to RAN, which causes GDP to disassociate or to leave RAN's binding pocket. And then because there's a vacant spot, the GTP molecule will diffuse into RAN's binding pocket, which means RAN is now bound to GTP, which is great because it's active. Yeah. And so when that happens, you have RAN GTP, or RAN is active now, and RAN GTP is able to bind to important. And when RAN GTP binds to important, important is able to drop the cargo protein or release the cargo protein. And when that occurs, then RAN GTP and important together will leave the nucleus. And this is because important cannot leave the nucleus by itself because it doesn't have the NES sequence. That's why it needs the help of another protein to be able to leave the nucleus. Then you have the RAN GTP important complex, I guess, leave the nucleus and it's out in the cytosol. Once that happens, we need to be able to deactivate RAN so that it can let go of important, so important can go do its job of bringing more proteins in. And so the way that this process occurs is that the GAP protein will bind to RAN GTP, and when the GAP protein binds to RAN GTP, it'll assist RAN in hydrolyzing GTP to GDP. And so then you get RAN bound to GDP, and RAN bound to GDP is useless and inactive. And when RAN is bound to GDP, it no longer has good affinity for important. So then it lets go of important, and important ends up back here to do the cycle all again. Did that make sense? What's going on there? Okay, great. Any questions? Okay, I have a question for you. If there's a loss of function mutation in GAP, what would happen to nuclear import? So to be able to answer the question, you have to first figure out what is the role of GAP in normal nuclear export. Anyone want to try answering that? No? No? Okay. Well, GAP would still be there, just wouldn't be functioning correctly. But I think you're correct in that. I wrote out such a longer answer. You said it so simply, and I wrote it out anyway. Okay, so whenever I approach questions like this, like it helps, I think of it in steps. And so when people were doing this one on exam 1, I try to break them into steps. So first you need to establish what's the role of GAP in normal nuclear, is it typo in there? Normal nuclear import. So GAP would help accelerate the hydrolysis of GTP and GDP, and that's what helps RAN to become inactive. So that's GAP's normal function. And so if there was a loss of function in GAP, meaning that it's no longer able to perform its function, then that would mean that you would have RAN GTP that would remain stuck to important and accumulate in the cytosol. And then eventually that would mean all your importance would, and you have to imagine in the cell, there's hundreds of thousands of these cycles going on. So you have many importans and many RANs. It's not just like one cycle in a cell. There's like multiple nuclear import cycles. And so eventually after a prolonged period of time, all of your importans would be stuck to RAN GTP. And that would mean you wouldn't have enough importans readily, freely available to take cargo proteins in. And so that would like bog down nuclear import. Did that make sense? Good job, by the way. Yes, import would slow down and eventually, I don't know why I didn't type the rest. Anyway, what I said. Okay. What about GEF? What would happen if there was a loss of function mutation in GEF? What do you think would happen with nuclear import? These are just thinking questions, you know, problem solving, LoRa type approach. Okay, nobody wants to answer, I'll answer. So GEF helps RAN to exchange GDP for GTP, which causes RAN to become active. If GEF is not functioning properly anymore, or it doesn't do its original function, then RAN would never ever be bound to GTP, right? Because RAN, the enzyme, can hydrolyze GTP, but it can't go the opposite way. It can't go from RAN GTP to GTP. So as a result, you just have RAN bound to GDP, and this cycle would be around here, this point. So RAN would always be bound to GDP, and if RAN's always bound to GDP, then there's no way for us to bring important like out of the nucleus, and all these steps here would be messed up. And no RAN GTP means important cannot leave the nucleus. And this question highlights that RAN GTP is necessary for the pathway because it brings important out of the nucleus. And so without that, you're stalling nuclear import. And so, yeah, so hypothetically, the important cargo protein complex would be stuck together in the nucleus. And you wouldn't have any free importance here either in the cytoplasm because and probably all be stuck like in this step. Does that make sense? Question? The which? I can't remember. She sent an email like either today or yesterday. I've been bad at reading my emails, but she said the ones that were given on exam 1 would appear. But the other ones, yeah, the other ones, yeah. So exam 2, your pathways, guys. Please draw those, please. Some people forgot to do it for exam 2, and I just was like,
But you'll
please.
get these ones.
Yeah. So I think the amino acid sheet and this one, but please double check your email for that. Okay. Okay, nuclear export. Just gonna roll through what's going on here. So completely different than the other one, the nuclear import. So you start here in the nucleus. That's step 1. GAF. So GAF does the same thing. We know that. We know what GAF does. Okay, I'm just gonna. I don't know why it's animated. I animated this anyway. So then you have RAN GTP. It's active, and then what does it do? It binds to exportin. Wait. Hold on. No, it does. RAN GTP binds to exportin, and exportin is able to bind to the NES sequence on this cargo protein. And then when it happens, you have this special triprotein complex and they exit the nucleus. And then you have this thing here with GAP has the same role of accelerating the hydrolysis of RAN GTP, and then it becomes RAN GDP. And then, yeah, so then when that happens, RAN GDP can't associate with anything. So then the cargo protein gets released in the cytosol, which was the intended purpose of this entire thing, to bring a protein from the nucleus to the cytosol. And then you release your exportin too, so it can go back and do it again. But as you can see, the process is very different here because exportin is unable to grab the cargo protein without the help of RAN GTP, which is very different than import. So please note that. You guys will get these slides too, but I think these notes are fire to include on your cheat sheet. You wanted to include these notes. I would be honored. Anyways, so you can see the difference. There's import and export. These things are different. I just highlighted that. So yeah, any questions? Did that make sense for people? Can I get a thumbs up? Thank you, thank you. Okay, we're just gonna, I have practice question for export, but for time's sake, you guys can do that on your own and practice with the animations to check your answers. But we're almost there, guys. Last slide.
Okay, so now we're going to go over gene expression regulation in eukaryotes. So pretty much to go over like on an overview, of course, gene expression is the production of proteins, right? You take your DNA, transcribe it into mRNA, which then gets translated into an amino acid sequence, which then goes to your proteins. That's pretty much the path or the central dogma, right? And the only difference is that in eukaryotes, you're going to have your transcription in the nucleus and translation in the cytoplasm. But whereas in prokaryotes, right, there's no nucleus, so to speak. So that you're going to have both transcription and translation done at the same time in the cytosol. And that's going to lead to a few other differences that we'll go over. So again, the central dogma theory, you're going to go from DNA to RNA to protein. DNA is going to be read by your RNA polymerase to then turn into mRNA, which is your transcription. And then a ribosome then reads that mRNA to then know what sequence of amino acids to then tie together, which, as you know, then form a protein and its properties. So yeah, and like on this diagram, you have again your RNA synthesis or transcription, which gives you their mRNA, which then is translated into your amino acid, which again, if you notice, you have your carboxylic and amino acid end. So to go on, first you have you have a lot of like different points of that process where you can then have regulation. So your first one is going to be at the transcriptional level, right, to go from your DNA to mRNA. Your main one is going to be your general transcription factors, and these are pretty much proteins that are going to recruit or regulate where RNA polymerase can bind to, or which promoter region on it can bind to on the DNA, right? Because all RNA polymerase is non-sequence specific, which is important because it's going to need to be able to transcribe any region of DNA. But on the flip side of that, it doesn't have any sequence to then like target on, like, okay, which promoter am I going to go on? So that all depends on the transcription factors. So if you don't have a transcription factor, then you're not going to have any RNA polymerase activity. So that's one point of regulation where transcription factors are going to go. And again, these transcription factors are gene specific or sequence specific. So they can only get targeted towards certain promoter regions. For example, like with the trip operon, like we're going to talk about later, you're going to need your transcription factors to first get on the operon, and then you can have your RNA polymerase get recruited. And you can have this go in either direction. You can have positive enhancement, which is pretty much upregulating the activity of RNA polymerase, or you can have the opposite, negative, or inhibition or suppressing RNA polymerase. So that's again your first point of regulation. Any questions? And then going on, you have also your, again, remember that transcription happens in the nucleus, right? It's going straight from the DNA. And in eukaryotes, our DNA is going to be packed in chromatin or histones that are going to wrap around DNA. And that's another form of regulation because you can either have this chromatin wrapped either in a euchromatin form, which is when it's open. So it's physically going to be spaced out, and RNA polymerase and all these transcription factors can physically get into the DNA and attach onto the promoter and then start transcription. Or you can have it in its heterochromatin conformation or its closed state, which is then, as you can see on the top, all the chromatins are packed too tightly for any proteins to get in there. So that's your second point. And both of these, transcription factors and chromatin packaging, are going to be at the transcription level or inside the nucleus. Going on, we have then your second point of where you can have regulation, which is your RNA modification or processing, right? This is either right before you have your mature mRNA or right after it's processed, but before it's like taken out of the nucleus. So RNA needs to be processed and made stable into mature RNA in order to leave the nucleus. Why? Because A, the cytoplasm, the conditions in there are very good for having a single stranded mRNA, and also because the cytoplasm has a bunch of nucleases that go around that are going to chip away immature mRNA. So pretty much how do you make a stable mRNA, or how do you mature one? You pretty much just add a 5' guanine cap on one side, on the 5' side, and then you're going to have your poly A tail or your adenosine tail on the other side. And that's going to serve two functions. One, you're going to have the 5' cap just to covalently bond it to the 5' end. And that's pretty much going to act as a buffer, right? Like we mentioned, there's a bunch of nucleases. You have your exonucleases, endonucleases, and their job is just to cut up extraneous mRNA, right? Because again, like the cell wants to be, again, like the cell wants to be as efficient as it can, so it doesn't want any extraneous, pieces of mRNA floating around. So if nucleus does find that, specifically an exonuclease, it's going to chip away at the 5' or the guanine cap and leave the actual mRNA untouched. And on the other side, you have your 3' poly A tail, and that's going to allow the mRNA to be able to leave the nucleus and into the cytoplasm. So that's your second point of regulation. You want to mature the mRNA strand that just got transcribed. That's good. And then more on RNA processing, you're going to have splicing. Again, this is only in eukaryotes. You have alternative splicing. So pretty much your immature mRNA is going to have these sequences of non-coding RNA, which are your introns, right? These sequences don't have any protein that they can form or any amino acid. So what the cell is going to do is it's going to cut out those areas and then just have your spliced mRNA, which is just going to be made up of exon regions. And the cool thing that in eukaryote cells you can have is you can alternatively splice this in different sequences. So let's say you have an mRNA sequence that has 3 exons labeled 1, 2, and 3. You could either have the cell put that mature mRNA together in that order, or you could have it going from like 2 to 3, 3 to 2 to 1, or 1 to 3 to 2. And that pretty much just increases the efficiency of how much protein or how much product you can get from one sequence of DNA. So yeah, and again, that's just, and all that's done by spliceosomes. So those are enzymes that are going to come in and cut out the introns. So yeah, is there any questions? That's going to be that's again going to be RNA processing. This is all in the nucleus of the cell. Okay. So now you have your post-transcriptional control, right? You have your mature mRNA. It has all the introns spliced out. It has just exons, and it has that 5 guanine cap on one side, and then the 3 poly A tail on the other. So now that it's gone out into the cytoplasm, you need to worry about its stability or how long it can survive within the cell. And that's going to be, again, maintained by it's going to vary by different mRNAs, but it's also going to be helped by that 5' cap and poly A tail. And we're going to go over later, but cells control the overall mRNA degradation as its own form of regulation. And like we talked about, RNA interference, such as like siRNA or mRNA, and also like mRNA localization, the cell controls which where the mRNA is going to be designated. So say if the cell has too much of one mRNA or doesn't need one protein as much, it's going to sequester these mRNAs to say P bodies where they can either be held in place or just degraded. So that's going to be your third point, your post-transcriptional control. Okay. And then your fourth one, translational control, right? This is going to be right before or right as a ribosome binds to that mRNA. So this is pretty much just controlling how the cell or the affinity for that ribosome for mRNA. So for example, there can be another protein that comes on that blocks that mRNA from getting translated, or the opposite, upregulates it. So, and again, why? So this pretty much controls that. Again, like mRNA is just floating around the cell, right? It's just by chance does it run into a ribozyme. So the cell can either increase that or decrease that likelihood. And that's pretty much with the end goal in mind of how am I going to control how many amino acid sequences I have, which pretty much leads to how much protein am I going to need or not have, or not need. Is that good? And then lastly, you have your post-translational control. This is going to be after translation. So it's mainly going to affect the amino acid sequence or the protein as a whole. So as you probably know from other classes, in terms of controlling the protein activity, you have allostery, right? So that's pretty much allosteric control is that you're going to have another protein that can either bind to part of the protein that is not the active site, but it's going to nonetheless change its conformation, which is then going to affect how good it is at its activity. So you can either have upregulation again or deregulation. Usually, if you want to activate a protein, you're going to add a phosphate for phosphorylation, or the opposite, you're going to methylate or ubiquitinate if you want to degrade the entire protein. And then you also have nucleotide binding, right? You have either GTP, GDP binding, or ATP binding, which is also going to affect the activity of a particular protein. And then lastly, you also have protein degradation, which is if you don't need a protein at the moment, you can just degrade it, which is carried out by either your unfolded protein response, right? If the protein's damaged in any way, say it gets degraded, then there's no use for it, and it's just ubiquitinate there. Or you also have ubiquitin mediated proteolysis, which is just tagging a protein for degradation inside of a proteasome. So is that good? Yep, and this is just a summary of all the different points of regulation, right? Again, you have your central dogma of DNA goes to RNA to mRNA to then protein. So right, you can either have control at the transcription level, or you can have it at the RNA processing level during the transport or translation or post-translation. And then you have prokaryotic gene regulation. And again, like the main difference here is that there's not going to be any chromatin control. There's not going to be any worry about exporting the mRNA. And also you're going to have transcription translation occurring at the same time. And here we have a table that summarizes the main similarities and differences. Again, the main differences are that in eukaryotes, our DNAs, there's going to be a couple extra steps because we have so many more genes and so much more compaction of that. So you're going to have to get around the chromatin issue, the nuclear issue, and you're also going to have transcription factors in eukaryotes versus sigma factors in prokaryotes, which pretty much just serve the same function. And the other important thing is in prokaryotes, you're going to have a lot of genes that are polycistronic. So one gene is going to lead to multiple different proteins, whereas as in eukaryote cells, you're going to have alternative splicing, which is just rearranging exons or getting rid of introns that don't have any product. But again, the similarities, in both cases, you're going to be regulating how often or how unoften mRNA gets degraded, where it gets localized, and then the overall rate of translation or production of your protein or product. So is that good there? And then, like we mentioned, sigma, that's going to be your transcription factor for prokaryotes. It's pretty much just, it recruits the RNA polymerases to its promoter region, and it's going to bind at 2 places on the DNA. It's going to bind at 35 down from the title box, and then at 10 down. And that's pretty much because at these points, especially at the -10 area, there's going to be a lot of A's and T's, and those have just 2 bonds between them versus G's and C's, which have 3. So it's just easier for the cell to break open the DNA at that point. And also, again, while sigma is sequence specific, it regulates that a lot. It regulates a lot of genes, so it attaches to a lot of points on the prokaryotic genome, and not with the same affinity. And that's where you have your consensus sequences, where your consensus sequence is like an average of all the places that sigma can bind to. But those individual places could have different affinities, and that just makes sense because, say, if you have housekeeping genes, you're going to want sigma attached to that with a lot more affinity because those genes are essential to the organism's like overall like health, versus like if you have like smaller genes that you don't really need on all the time, then sigma doesn't need to be as strongly binded to those areas. So is that good? And then
I know it's 9. Yeah, so I know it's 9. So I'm gonna try to just, pick out the key points, main topics, or like essential points. If you guys have any like questions along the way that I'm not touching on, like definitely stop me, let me know. But otherwise, we're gonna try to go on through. Okay, cool. So operons in general. Each operon contains regulatory DNA sequences that act as binding sites for regulatory proteins. So there's 2 parts that some students might get confused on. So there's a promoter and then there's the operator. The promoter is where the RNA polymerase binds to, but then the operator is the repressor binds to. And that kind of makes sense if you think about it, because if the repressor is in front of the promoter and it's bound, then the RNA polymerase can't go forward because it's blocked by the promoter. And operons in general can be inducible or repressible. Specifically for the TRP operon that we focus on in this class, it's repressible. So that means that usually the TRP operon is on, so the cell is making tryptophan, but when it needs to be, it gets turned off because of a specific molecule. In this case, when tryptophan is present, it gets turned off, so it's repressed. All right. So looking more specifically at the TRP operon, TRP stands for tryptophan. It's a type of amino acid. There are generally 3 case scenarios that can happen. One is when you have low TRP levels. That means that the cell does not have access to tryptophan, so it doesn't have a lot of tryptophan around. So that means it, of course, has to make more tryptophan. So your operon is going to be on because it wants to make more tryptophan. However, when you have high tryptophan levels, that means there's a lot of tryptophan floating around. It wants to conserve energy. And so the TRP operon is going to be off so that it's not making any more unnecessary tryptophan that it doesn't need. And because of this, so is the TRP repressor protein a positive or negative regulator? And the answer is that it's a negative regulator, because when the TRP repressor is bound, it's going to negatively regulate, or it's going to stop the production of tryptophan. This is just a video of like TRP operon. Some people are more visual learners. So if you have the time, or if you want to later on for like a better, clearer picture, definitely go back and watch this video. I know it's kind of fast paced. So again, like if you want me to stop at any point, let me know. All right, so this is like a general overview that I think some TA a while ago drew. Very helpful. But this is a trip repressor. And so when the trip repressor is not bound to the tryptophan, it is not bound to the operon. So this entire operon is on. However, when tryptophan is around, so again, like high levels of tryptophan, it will bind into the binding pocket of the repressor. The repressor will then be able to bind to the operator, and then it'll be blocking the RNA polymerase from doing its job, and you will have no transcription of all of these genes. Does that make sense? Like the overall picture of how it works? Cool. So then we'll get into specific scenarios that can happen. So again, scenario 1 is high levels of tryptophan. When you have high levels of tryptophan, that means there's a lot of tryptophan around. So if there's a lot of tryptophan around, that means it's more likely to be able to bind to the repressor. So the tryptophan is bound to the repressor, and then that means the repressor is active. If it's active, it's able to go and bind to the operator. And when tryptophan is bound, or the tryptophan repressor is bound to the operator, the RNA polymerase is blocked from being able to bind to the promoter. And if RNA polymerase cannot bind, then you can't even initiate transcription. So the entire operon is off. Sounds good? Awesome. So the next couple of scenarios requires understanding of what attenuation means. So just a little overview, attenuation itself means the early termination of transcription. So essentially you can think of it like the prokaryote might start to transcribe, but then it realizes that it might have made a mistake or it wants to conserve energy. So it's like, just kidding, let's backtrack. It terminates transcription early. So attenuation sequences is before the DNA. So your attenuation sequence, I think if you see it here, is right here. So if these are all of the parts of the DNA that actually makes tryptophan, attenuation is the part of the DNA before all the trip DNA. And there are 4 regions in this sequence that can pair with each other. So regions 1 and 2 are complementary. So that's the blue and the pink one. Regions 2 and 3 are complementary. And then regions 3 and 4 are complementary. 1 and 2 means pause. 2 and 3 is what allows for continuous translation and transcription. And then 3 and 4 is what actually causes attenuation or termination. And this can all happen because prokaryotes are able to transcribe and translate simultaneously. And that'll make more sense in the specific scenarios. So scenario 2 is low tryptophan level. So if you don't have a lot of tryptophan around, that means it won't be able to bind to the repressor. So the repressor is inactive and it won't be able to bind to the operator. If it's not bound to the operator, then the RNA polymerase can bind to the promoter. And then that means RNA polymerase can start transcription. There's, of course, no attenuation because RNA polymerase is just working normally, doing its job. So a little bit more specifically about like how the attenuation sequence works for this. So first, when your RNA polymerase binds to the promoter, it's going to start transcribing, doing its thing. It transcribes regions 1 and regions 2. And of course, because there is simultaneous transcription and translation, as the mRNA strand is coming out of the RNA polymerase, ribosome can go ahead and bind and start transcribing. And so then once the ribosome binds, once regions 1 and 2 come out of the RNA polymerase, they are able to base pair with each other because they're complementary. And that's what causes a pause. And by pause, it's the RNA polymerase that's pausing. And so that just lets the ribosome some time to catch up to the RNA polymerase. Once it catches up, it's going to knock down regions 1 and 2. So if you can kind of picture like the ribosome is going, the RNA polymerase is paused, ribosome goes, reaches this little loop, and it's going to knock it down because ribosome can only translate a single-stranded mRNA. So then it knocks it down, and now it's flat. But once the 1 and 2 loop gets knocked down, RNA polymerase is able to start transcribing again. But this is a sequence where the ribosome can get stuck, so to say, because there's not a lot of tryptophan around. So when the ribosome gets to this codon sequence, it has to wait for tryptophan, the amino acid, to be able to like go into the ribosome to be added to the to be added to the polypeptide sequence. And so if there's not a lot of tryptophan around, it's going to be stuck there waiting for a long time before any tryptophan comes. And so that's what allows 2 and 3 to bind together. And once 2 and 3 is bound together, that means that 3 cannot bind with 4. And if 3 cannot bind with 4, there's no attenuation. Does that make sense? Any questions about how it works? Okay, yeah.
And if there's no tryptophan, which is like waiting, whatever.
So if there's no, so it's just that usually there's no like no tryptophan at all. That's why the scenario is like low levels of tryptophan. So it just takes a lot longer for tryptophan to get to the ribosome because there's not a high concentration. But eventually it will go again once the tryptophan makes it. It's just stuck there, not permanently, but like for a long time, or at least long enough for 3 not to be able to pair with 4. Does that make sense? Cool. So then the last scenario is low to high, or like the medium scenario. So this means that initially tryptophan is not bound to the repressor. So you have an inactive repressor because it's not bound to the operator. Again, that means RNA polymerase can bind to the promoter and start transcription because the cell thought that at first there's not enough tryptophan, so they have to go make more. However, later on, the cell might realize that there actually is enough tryptophan present. And if there is enough, it's like, why do I have to waste my energy making some tryptophan that I already have? And so the solution is attenuation. Okay, so the mechanism for attenuation is, again, the same thing. RNA polymerase goes through, it creates 1 and 2. 1 and 2 comes together and it pauses to let the ribosome catch up. Same thing as before. However, because now there is a lot of tryptophan around, when the ribosome is on region 1, it's not going to get stuck on region 1. Instead, it'll be able to move on fairly quickly because there's a lot of tryptophan around to just kind of go in and be added to the polypeptide. And so instead of being stuck on region 1, it's going to keep on going until it gets to the stop sequence right before region 2. And when it's stopping right before region 2, it kind of like overlaps onto region 2. So that means it's blocking region 2 from being able to bind to region 3 like it would in scenario 2. And that means that 3 is open and available for 4. And when 4 and 3 bind together, essentially it like snaps together quickly and yanks it out of the RNA polymerase, what's it called? The active site, the RNA polymerase active site, because it's A and U rich. So it's not that strong. It's fairly easy to break all those bonds inside the RNA polymerase active site. So key note here is that instead it blocks region 2 and then 3 and 4 can come together. That yanks it out of RNA polymerase, and therefore RNA polymerase can't finish transcription. Does that make sense about attenuation? Okay. This is just a little summary, like compacted version. Some people like to see it this way, so it's just like another way for people to study if they need it.
Okay, real quick. How many of you raise your hand are familiar with the game Kahoot? Yes? Okay. So to reward you guys for sitting through this review and powering through, the next review on Sunday night is going to be a Kahoot quiz game hosted by yours truly and David. I still have to make the questions. But if you beat my Kahoot, I think maybe I might give out a nice prize. Maybe free swipes and grab and go, or food or something. Maybe a gift card of some nature, whatever. But the next review session is Sunday night. Sunday night 6:30. So come with your smart enabled device, you know, laptop, tablet, all that jazz. But anyways, real quick, just wanted to say for our internet folks watching, we have hyperlinks for slides from way back then. So if you need to review weeks 1 and 2, you need more detail, click the links, you go there. This one is the special domain subunit slide I was talking about earlier. Explains it very in depth because this is a tricky topic, I felt like. And then weeks 3 and 4, all the recordings for YouTube, the links in their slides because we're nice like that, all the things included. Okay, and that's it. Thanks for coming. Appreciate you guys powering through.
We know it's really long.
We know it's really
Thank
long,
you.
but next time there will be a Kahoot session and it's gonna be a fun time.